Good afternoon, good evening, everyone, and welcome to the final session of today's DDF Summit. Next up, we have Dr. Angus Forster, the Chief Development and Operations Officer at Vaxus. Dr. Forster's worked in medical product development and commercialization for 20 years and has overall responsibility for the research, development, and operations of Vaxus. Since 2012, he's established the Vaxus R&D team that includes more than 70 scientists, engineers, and professional staff, and led 3 phase 1 clinical trials. He brings considerable experience in pharmaceutics, formulation development, analytical science, medical device design, and development, risk management, and quality systems. Before joining Vaxus, Angus commenced his career with GSK in pharmaceutical development, spent 5 years with PA Consulting in the UK, working on medical product development projects, and has a PhD in pharmaceutics and a BPharm honours, both from Otago University in New Zealand. With that, I'd like to hand over to you, Angus. Please go ahead.
Hello, I'm Angus Forster, and I lead research and development at Vaxus, a company developing technology specifically designed to improve vaccination, immunologically, logistically, and for the patient. I'd like to thank the organisers of the DDF Summit and the session leads for providing this opportunity to me to present our technology and its intended benefits, but also to discuss challenges around formulating vaccines in the dry state for delivery into the skin. In this talk, I will cover the potential benefits of skin delivery of vaccines with high-density microarray patches and go through our recent supportive clinical data. I will then dive into the formulation of the vaccine and the role of the excipient, Captisol, in combination with the seasonal influenza antigen and how the excipient was selected. I will also touch on a published study that focused on formulation of an activated poliovirus vaccine, IPV, using the same technology. This is how we see the future of vaccination. Apply to the skin, press the button, wait 10 seconds, remove. That's it. It's a better scientific approach that can dramatically improve a country's ability to respond to a pandemic, and it's also a great way to improve the reach of vaccination for a broad range of infectious diseases. So what do we have? High densities of immune cells live just under the skin surface, in the epidermis and upper dermis. Our technology uses a small polymer patch, shown lower left, that has thousands of very short microprojections with high density across the skin contacting surface. Each microprojection is around 300 microns in length and just breaches the stratum corneum. We use our patch to deposit vaccine right amongst the immune cells in the skin, where they can be rapidly brought to the lymph node. We dispense liquid vaccine formulations onto these projections, as shown in the upper right picture, using a technology developed in-house that allows accurate deposition of picoliter-sized drops to many projections at once. These discrete small drops also dry very rapidly, and this rapid drying process of the vaccine in combination with excipients has been shown to lead to improved thermostability. The needle and syringe has been an effective way of delivering medicines since around 1853, but as we all know, it has significant drawbacks and complexity and is not typically preferred by patients. It is also an especially challenging delivery technology for use in pandemics and for enabling vaccine access in low and middle income countries, requiring skilled administration and maintenance of cold chain. Our hypothesis is that by removing the needle, targeting the abundant immune cells in the skin, and drying down the vaccine, that we can transform the process of vaccination and improve vaccine effectiveness. It's important to note that we are still a couple of years away from completing clinical development and scaling up manufacture to supply the quantities of product that are needed in pandemic response or for routine vaccination, and scale-up remains one of the most significant challenges to adoption of microarray patches for vaccination. But before I talk about formulation, at the center of the process, and of course the finished product, is the applicator, which combines a vaccine-coated HD map within a single-use disposable applicator system. The applicator delivers the vaccine to the skin consistently, removing most of the user dependency, and has been designed to provide feedback to the user that the dose has been administered. The applicator can, with associated desiccant, also packages and protects the sterile product from contamination and moisture ingress. The device elements have also been designed for logistics, both as components and as a finished product, to reduce unnecessary packaging and cost, but also designed for manufacture. The body of the applicator is made from aluminium, and the part count is kept low. We have aimed to make the total cost of the goods, excluding the vaccine, at least comparable to, and potentially less than a pre-filled needle and syringe. So what's the major barrier to adoption of these technologies? In our view, the biggest challenge to MAP developers, and the reason why the needle and syringe is such a tough competitor, is scalability of manufacturing and cost of goods. We invested early in developing a finished product and manufacturing process that can enable high volume, low cost aseptic manufacture of finished product. Our process is based on sterile incoming components, the applicator and the MAP, disposable technology for dispensing vaccine, and a focus on developing in-process testing to assure the quality of finished goods. We've developed in-process tests to determine where the vaccine formulation goes on the MAP when dispensed, and feedback loops from the dispensing system to help quantify volume dispensed. We are not done yet, but in combination with German manufacturing automation specialist Harro Hoflinger, we are de-risking key process steps in Australia and in Germany before finalising the pilot design and commencing the build. We have a number of well-funded programs with leading partners that we're currently working on. Programs with the Gates Foundation on measles and rubella to improve access and vaccination rates in countries where children are still suffering from a vaccine-preventable disease. A collaborative project with Merck, a recent project with the US government in pandemic influenza response, but also other programs across different infectious diseases and cancer vaccines. So this number of projects keeps us very busy, and the team at Vaxis is currently over 70 staff working across scientific, engineering, and other disciplines. We are not the only ones who think this approach to vaccination is an important development and opportunity. The Vaccine Innovation Prioritisation Strategy aims to drive vaccine product innovation to better meet country needs for immunisation coverage and equity, by providing clarity to manufacturers and partners for investment decisions related to vaccine innovation. This prioritisation strategy recently concluded, recommending microarray patches as the first choice for vaccine innovation. It is also clear that MAP technology can also meet the aims of priorities 2 and 3, providing heat-stable and controlled temperature chain vaccines, and is well suited to barcoding and traceability. One of the defining features of our MAP technology is the high-density microprojection array, which we think offers benefits for improved immune responses. Due to the high density of microprojections, we need to apply the MAP to the skin with sufficient speed to enable penetration of those microprojections through the stratum corneum. This is achieved via the use of an applicator device containing the MAP, as shown in the previous video. When the applicator is triggered, the MAP flies at around 20 meters per second for a very short distance before engaging with the skin. Published work has shown that this dynamic application results in cell death at the application site, which is significantly greater than with ID injection. This damage event creates alarm signals, damage-associated molecular patterns, in combination with proximal delivery of the antigen from the high-density array. This co-localization of vaccine, cell death, and alarm signals is a hypothesis for why we've seen significant dose sparing in preclinical models. Showing improved immune responses in models is great, but we've now shown the benefit in clinical studies. We've published data from 3 clinical trials run in Australia, the most recent of which was the largest MAP vaccination study run to date, and also the first to look at reduced vaccine doses delivered by MAP in comparison to standard doses given by needle and syringe intramuscular injection. We also have a phase 1 study ongoing at the moment with live attenuated measles and rubella virus. But first of all, I'd like to share some of our data from our Singapore influenza clinical study. This study used an H1N1 egg-derived split influenza antigen. At doses similar to needle and syringe, shown in grey, we saw statistically higher functional antibody titers using our HD map when delivering to both the upper arm site over the deltoid muscle, shown in blue, and the volar forearm, shown in red. We saw higher titers at day 7, day 21, and out to day 60, which was the final time point of the study. The biggest increase in titer compared to needle and syringe was seen at day 7 time point. We also took the HD map delivered dose down to 1/6 of a standard needle and syringe dose, down to 2.5 micrograms of functional HA protein delivered by MAP in comparison to 15 micrograms given by intramuscular injection. As you can see from the graph, at this lower map dose, our functional antibody responses, shown in green, overlaid those with the higher dose IM and were maintained out to day 60. We have more work to do with additional seasonal influenza strains to show that this performance is product relevant, but it was an encouraging first clinical result at lower doses. The ability to use lower vaccine doses could be hugely beneficial in a pandemic vaccine rollout, as shown in the table, in terms of making more doses of vaccine available more quickly. As part of the same clinical study with the H1N1 influenza antigen, product was placed on stability for up to 12 months at a range of temperatures, 2 to 8, 25, and 40 degrees centigrade. Our dried vaccine product showed no significant decrease in HA potency for 12 months at all conditions, whereas significant losses in potency occurred after 1 month at high temperature for commercial liquid vaccine. The vaccine coating on the microarray patch, as shown in the scanning electron micrograph image, showed no degradation on storage. This type of stability performance opens up the opportunity for different models of vaccine storage and distribution. In the next part of this talk, I want to focus on the vaccine formulation used in this study that achieved such great stability performance. I also want to run through the approach to selecting excipients that are compatible with the HD map and the manufacturing process, but that also confer the necessary performance required for the technology. Our initial work with seasonal influenza commenced with the A California H1N1 antigen before migrating across to A Singapore due to a change in the selected seasonal H1N1 strain. This change in antigen type with seasonal flu vaccines also means formulations used to stabilize drug product need to be transferable across different influenza virus strains. Within the supply constraints of seasonal flu vaccines, there is insufficient time to undertake extensive formulation development between strain selection and supply of commercial product to both the northern and southern hemisphere markets. For the H1N1 strain, which was a similar virus and HA protein structure, we had no issues adapting the initial work with A California to the A Singapore antigen. The antigen we received was in a more concentrated form than the typical commercial seasonal flu antigen and provided in a lower salt buffer. To identify excipients that would confer the necessary stabilizing and process-compatible properties to the formulation, we worked with Professor David Volkin's group at the University of Kansas. This screening process, which had elements unique to our product, generated a number of potential excipient hits for stabilizing the A California antigen. We further down-selected at Vaxis based on other product and process attributes, resulting in the selection of sulfur-beta-ether beta-cyclodextrin or Captazole as our preferred excipient. The overall formulation pie chart showing the HA protein content, active component, additional total protein, buffer salt, and Captazole ratios is shown on the right-hand side. The ratio of SBECD to HA protein was 4:1. The pie chart excludes an expected low residual moisture content. Firstly, before I talk specifically about the attributes required of excipients used in combination with the vaccine and our technology, I would like to describe our method for dispensing liquid vaccine formulations onto the HD map. The technology is based on small drop dispensing, with individual drops targeted to dispense onto individual microprojections. Multiple drops are applied to each microprojection following a short drop drying period to load on the required dose of vaccine. Our first embodiment of the technology used a single dispensing nozzle built by Biofluidics, which was aligned to the map and dispensed a drop to each microprojection in turn, moving up and down the rows of microprojections before returning to the origin and repeating this process. This technology is still used in-house for early stage studies. To speed up the dispensing process, we have scaled up to dispensing a quarter of an entire map array with a single pulse onto a square map, using a multi-nozzle dispensing system called the MJet for current clinical work. However, we are also now achieving a full circular array dispense with a single pulse, jetting up to 3,000 individual drops lined up to each microprojection. The array and mold insert for the HD map are made using the same fabrication process, and the software aligns the printhead containing the nozzle to the map. The next video shows our proof of concept line for performing this step. In this video, you will see a screenshot of the rapid image QC analysis of each map positioned onto the applicator top. The vision system checks for microprojection position, feeding this information directly to the dispensing head, which makes small adjustments to align the nozzle array to the exact position of the microprojections. This image analysis can also detect defects in the map. The printhead dispenses one drop to all 1600 microprojections on the array simultaneously, and the video shows an output of 200 parts per minute. So you can see the camera system taking an image of the patch as shown on the left hand side, which collects that image, feeds in the information about the projection position to the printhead, which you can see there on the right hand side, making small adjustments to line up the nozzle array to each microprojection on the patch. Along with being compatible with our dispensing technology, any added excipients to the vaccine formulation need to exhibit critical attributes, either alone or in combination. The Kansas University team used a library of excipients, mainly from the FDA's inactive ingredient list, for use in injectable or ocular products. The excipients need to be highly aqueously soluble to be compatible with the vaccine buffers and also to ensure that high concentration excipient stock solutions can be prepared. The key feature and major component of the screening approach is to identify excipients that benefit vaccine stabilization as a liquid vaccine coating solution, but more critically, during the rapid evaporative drying process and on subsequent storage. The skin delivery properties and dissolution kinetics are also critical and to an extent assessed in the screen by determining the extent of elution off the patch back into buffer. The Vaxxis HD map is designed to be on the skin for a short period, ideally 10 seconds or less, to deliver the required dose into the top layers of the skin. In the micrographs, you can see the removal of dried material on projection tips following application to the skin. Also, if an excessive amount of excipient is required to exert these effects, this can lead to issues with the amount of solids dried onto the HG map, which affects dissolution, but also the morphology and skin delivery performance. Excess solids leads to broadening of the microprojections and blunting of the tip for skin penetration. The dispensing technology used to coat small droplets onto microprojections is not simple to transfer to a partner at this stage and requires development to ensure liquid compatibility with the dispensing approach. In collaboration with Kansas University, we developed a simple screening rig that uses evaporative drying with dry nitrogen gas to rapidly dry small volumes of antigen and excipient solutions onto plastic discs in a well plate setup. The plastic discs are made from the same polymer resin as the HD map and go through a similar molding process. This enables many formulations to be tested as replicates in a screening format that is representative of the intended clinical or commercial dispensing and drying process. A short list of around 53 excipients in different classes was selected by the KU team for initial screening using the concentrated A California antigen. The steps involved in the screening process are shown on the slide. First, the antigen alone was dried onto the plastic disc to determine whether it could be immediately recovered, eluted, back into a small volume of buffer. For flu, the core assays used were total protein by BCA and an enzyme immunoassay, EIA, specific to functional HA protein, developed by scientists at Seqirus as a more robust alternative to single radial immunodiffusion assays used for influenza product release testing. The total protein assay ensures that all dried protein elutes off the disc and informs of potency reconciliation via EIA. Once these steps were in place, a short stability screen on dried HA protein was performed to generate a condition that rapidly led to degradation of functional HA. In this case, in the dried state, the HA was quite stable, and therefore a 48-degree condition for 7 days was selected. And then excipients were selected alone in combination with the antigen. Results from the initial screen are shown in this graph, in this case for EIA potency. A similar set of data was obtained for total protein recovery. Potency performance was compared to freshly thawed antigen stock as a standard, in this case with some light sonication to disperse minor aggregations seen at these high protein concentrations. A comparison was also made to antigen dried down without excipient, which in some cases performed better than excipient combinations, and this is shown by the blue arrow. From this initial experiment, 5 lead excipients were selected: sucrose, aspartic acid, lactose, arginine, and capsazole. These 5 lead excipients were further screened at a range of different concentrations, keeping the percent weight per volume fraction low in order to restrict the amount of solids that needs to be dried onto the HD map. The formulations were taken out to 28 days, and both the total protein recovery and EIA recoveries are shown on these two graphs. 3 excipients were identified that provided the best recoveries at day 28: sucrose, arginine, and captazole, all of which maintained HA potency to greater than 60% depending on the mass fraction used. Not shown here, a further combination screen with these 3 excipients was performed, but this only showed a marginal benefit to HA recovery. Therefore, 3 formulations were transferred into Vaxxus for testing using our in-house dispensing approach. Based on a combination of dispensing stability and droplet targeting, captazole was selected to move forward into clinical development with ACE Singapore. As mentioned, the first example used an antigen which had a reasonably good thermostability without excipient in the dried state, with the screening temperature needing to be increased to 48 degrees centigrade to rapid screen for an excipient stabilizing benefit. An activated poliovirus vaccine, or IPV, represents a much more challenging case, so I'd just like to spend a few minutes discussing the results from a similar screening study, the results of which have been published in the Journal of Pharmaceutical Sciences. The move to work on a microneedle or MAP delivery of IPV was a response to the switch from live attenuated trivalent or bivalent oral polio vaccine to IPV to mitigate small risks of vaccine-associated paralytic poliomyelitis and outbreaks of vaccine-derived polioviruses. However, IPV is more expensive to produce, is typically given by intramuscular injection, requiring skilled healthcare workers, and the injectable requires cold chain storage to prevent potency loss. In collaboration with the World Health Organization, we worked on a MAP IPV product. Other studies had looked at dried IPV formulations, freeze drying, spray drying, vacuum drying, and IPV types 1 and 3, especially type 3, are known to be problematic to stabilize during dry down. A similar approach to the work with influenza was taken, but rather than 48 degrees centigrade, a 25-degree accelerated condition was selected to screen excipients, using the type-specific de-antigen ELISA to determine potency of each type in the combined multivalent vaccine bulk. When used alone with IPV, certain excipients demonstrated a stabilizing effect on IPv3 during drying and on storage, in this case only 7 days at 4 degrees C. The bars represent potency loss, loss on drying in black, and loss on storage in grey. With the lower the bar, the greater the stabilizing effect. For IPv2, in comparison, almost all excipients mitigated potency loss on drying and storage. From that first screen, a combination of a reducing agent, amino acid, cyclodextrin, and gelatin were tested before down-selecting further to a cyclodextrin amino acid or a cyclodextrin reducing agent combination. The data shown in the slide shows a potency of IPv3 and IPv2 when dried and stored for up to 3 weeks at 25 degrees centigrade, showing improved stabilization of IPv3 and a relatively stable performance of IPv2. However, for both types, without added stabilizing excipient, potency is essentially lost on evaporative drying after 1 week at 25 degrees C, as shown on the far right of each graph. It was clear, for type 3 in particular, that even with stabilizers, there is significant potency loss over 3 weeks at 25 degrees centigrade. A key question was whether this loss stabilizes or would continue to increase significantly on further storage. Therefore, the top 2 performing stabilizing formulations were then tested to more fully understand the kinetics of stability over a short period, with daily de-antigen ELISA testing over 7 days and then at 14 days, to see if the rate of potency loss stabilized after an initial hit on drying and then subsequent storage at 4 degrees and 25 degrees C. The 3 graphs show this performance for each type in the multivalent bulk compared to an unformulated control dried in DPBS. The 2 formulations selected combined 2 different cyclodextrins in combination with reducing agent glutathione. Both formulations perform similarly on storage and significantly better than the unformulated material. The profile of loss indicates an apparent leveling off, and therefore a smaller study with Formulation 1 showed stability of the product out to 52 weeks at 4 degrees with only a 20 to 30% potency loss. The required combination of excipients indicated a potentially different role for these excipients in stabilizing IPV during drying and subsequent storage. We've now run 3 vaccine stabilizing screens with Kansas University, 2 of which have been published. We've seen positive stabilizing results with excipients for all 3 vaccines: influenza, a split protein vaccine, IPV, and whole inactivated virus vaccine, and a live attenuated virus vaccine, measles and rubella, which I've not discussed. With measles and rubella, there was no clear benefit to the addition of Captisol, but this was done in the background of gelatin present in the virus bulks and used to stabilize the freeze-dried vaccine in the commercial product. Removal of the gelatin component is not straightforward, and therefore the benefit of the screen was limited to determining which additives may further improve dry down stability in combination with gelatin. To discuss the mechanism of action of stabilization for flu and IPV antigens, which is by no means completely understood, I want to finish the talk by discussing the structure of Captisol. Cyclodextrins are cyclic oligosaccharides, presenting a hydrophilic outer surface and a lipophilic core. This can contribute to macromolecule stabilization, most likely through masking of hydrophobic regions and decreasing aggregation, potentially through hydrogen bonding, limiting protein mobility. In our example, with rapid evaporative drying, the local environment around the protein changes remarkably: local pH, tonicity, material interactions, and therefore use of a stabilizer that can inhibit interaction and protect the antigen and vaccine during drying is critical. From the stabilizing work conducted with KU, we are building up a library of stabilizers that work with different vaccine types and are compatible with our dispensing technology. We are also using Captisol as an excipient in additional vaccine development programs, either alone or in combination with other excipients. And finally, I'd like to acknowledge the team involved in the A Singapore clinical study, especially in the immunological assays and supply of materials, as well as analytical testing, but also the KU team involved in the IPV and flu work. And finally, I'd like to thank you for your attention during this talk, and I'm happy to take any additional questions during the live Q&A session.
Thank you very much for that, Angus. That was a great talk. So, Angus, I have a few questions here ready for you. First of all, what is the timing to see an HD-MAP vaccine product registered, do you think?
So where we are now at the moment, I think we're still completing phase 1 studies, but I think importantly we're scaling up product manufacturing. So I think the key barrier to adoption of these technologies is the ability to scale at high volume, low cost. So we'll advance in that area. I think what we've seen obviously in the last 18 months is a huge change in the rate at which vaccines can be registered. So I mean, I think previously we would have said, you know, at phase 1 stage, you're at least 3 to 5 years away from having a registered product. But I guess depending on the vaccine and the product that we're working on, that could be, you know, significantly shortened in today's climate. So we've seen that need to sort of move into phase 3 studies with a high volume manufacturing process that is representative of commercial volume to be the key barrier. And I think so. We're working with the US government at the moment on a pandemic influenza program, obviously hopefully preparing ourselves to be ready for the next pandemic if there is one with flu. So I guess if that was to be the case, then we could, if our manufacturing process is advanced, we could rapidly move to scale. With other programs, I think we're around about sort of 3 to 5 years away from market.
Okay. Question here in terms of the applicator. So question was saying why 10-second applicator time, and would that vary by product?
Yeah, great question. I think this also comes down to the nature of the formulation that we dispense onto the top of the micro projections. So our micro projections, as I sort of described in the talk, are solid. So they go into the skin, they deliver the coded vaccine into the epidermis and the epidermis. So we are reliant on a combination of properties of that formulation for that vaccine dose to be delivered into the skin consistently. So one of those is obviously the dissolution rate of those excipients and formulation as it is delivered. So that's why we use sort of highly soluble materials to improve that dissolution rate. The other thing you can imagine with all these micro projections going into the skin with a high density array, there is a degree of sort of stripping of those that coating as it goes into the skin and on removal of those micro projections from the skin as well. So that drives the drug sort of vaccine release properties into the skin. So it's a combination of dissolution and physical removal of the dried on coating as it goes into the skin. We think 10 seconds is a short period of wear time. Our previous clinical studies have actually had the product on the skin for up to 2 minutes to enable the complete delivery of the dose. But we've done further clinical studies without vaccines, which has shown that dried coating can deliver very rapidly into the skin with no variance in the amount of a marker delivered into skin with short wear times. Obviously, from a compliance perspective, especially if you're looking at a product which may enable less skilled healthcare work administration or potentially self-administration in the home in certain situations, obviously a simple procedure which doesn't require the person to count up to 10 minutes or 5 minutes of sort of residence time with the product on the skin would be very important for compliance. So obviously driving that time down to as low as possible with a degree of tolerance is important for different products.
Yeah. So on that front, in that 10-second delivery, how much residual antigen remains on the device after it's been removed?
So in our first studies, we sort of were delivering around about 60% of the applied antigen on the patch, and this is one of the sort of challenges, or another challenge with our technology is making sure we utilize the vaccine effectively. And what we have seen in preclinical studies is you need much less antigen to generate similar immune responses in preclinical models. And our recent study in flu showed that with 1/6 of the vaccine, we could generate comparable responses to a full dose given by intramuscular injection. But obviously, if we're not utilizing all the material which is dried down onto the patch, then we have a sort of an overage which we need to account for. But our what we call transfer efficiency, so that's the transfer of dried vaccine from patch into skin, is improving, and sort of recent sort of changes to the patch design and also the formulation have allowed us to get up to around about 80% utilization of the dried down vaccine onto patch, which is, I think, is pretty good compared to the overfill that you get in a conventional pre-filled syringe.
So following on from that, what drove the Microarray pattern design?
So if I go right back to the sort of the start of the technology, it was based on the idea of targeting directly into cells with vaccine delivery. So with the very high density arrays, just breaching the skin surface. And the original arrays had around about 21,000 microprojections per square centimeter. So I'd call them ultra high density arrays. There were some challenges there of actually trying to get that in a consistent manner into the skin. You can imagine it becomes like a bed of nails. So that hugely high density, sort of achieving consistent, tolerable penetration of those projections into the skin is a barrier. So that was the original hypothesis, that you would basically just deliver vaccine and antigen directly into immune cells in the skin. I think what we've seen is that and as I described in the talk, that combination of the energy required to get that breaching effect through the strand corneum, which does generate some local cell death, with the deposition of antigen and amongst that sort of milieu, if you like, of different signals, generates those improved immune responses. So we think that high density array has a benefit to immune response. It also enables us to achieve a decent loading of vaccine onto the patch because we have a higher surface area on which to deposit that vaccine onto, and we can modify that based on patch size and also density as well. So that was the sort of the starting point of the high density array, and we've seen that benefit through preclinical studies in terms of improved immune responses, and now in the clinic.
Great, thanks. Question here regarding the actual dispensing. So are they drops actually dropped down onto it, or is there physically brought together in order to transfer the liquid?
We've developed a sort of a picoliter size dispensing technology in-house. We started working with biofluidics with a single drop dispensing system. So we dispense drops that are around about 350 picoliters in volume. So they are very small drops. So we bring the micro projection tips in close proximity to the dispensing printhead. So there's an offset of around about 200 microns between the printhead and the patch surface itself. So that does require very careful alignment of the printhead to the patch substrate. But there's a degree of propulsion from the printhead. It's based on a piezo crystal, generating a pulse which drives the droplet through the nozzle towards the projection. And obviously you have to make sure that they align exactly to each micro projection. So that's been achieved now. We're running at quite high speeds on what we call our proof of concept manufacturing system, up to around 200 parts per minute in terms of coding process. Hopefully looking to drive it up to 600 parts per minute. But it doesn't rely on any sort of charging of the patch or any other sort of targeting performance, just purely the alignment of the dispensing, the nozzle array to the micro projection array.
Great, thanks, Angus. Just one thing again, just following on from that, in terms of the drying of those, Do you load more than one, or to go through the drying step? And how do you assess the dryness, if so?
Yeah, two great questions. So firstly, the drops are very small, the picoliter size drops. So even with just conventional drop imaging, if you have camera imaging the drop, you can see that reduction in volume as it's actually falling that small offset between the print head and the micro patch itself. We do look at different drying conditions, so we can use sort of an impinging drying gas to improve that drying rate. And you're right, to build up a vaccine dose, depending on the dose requirement of the vaccine, you do need to dispense more than one drop per micro projection. So one of the challenges is you have a degree of drying and then re-wetting, which can cause issues with vaccine stability. So I think obviously we've seen with different formulations that we can achieve very good stability during that dry down process. With certain vaccines, for example, the live attenuated virus, measles and rubella, you do get a loss in activity during that dry down step, potentially because of that re-wetting and drying step. Measuring the amount of moisture left behind on the patch is very, very challenging. So you've got, a coating of around 150 to 600 micrograms on a patch. So actually coming up with a method that allows you to detect this, probably about 2 to less than 2% of moisture is quite difficult. So what we end up doing is we actually store the patch with a desiccant, essentially a molecular sieve desiccant, which enables that, I think, final drying process and achieves probably a very, very low residual moisture content within the formulation itself. But we don't actually have a number for quantifying that amount of moisture. Previous data with different formulations suggested around about less than 2% residual moisture post drying, but that didn't include the storage effect of the desiccant as well to further decrease that moisture content.
Okay, brilliant. Thank you very much, Angus. Really appreciate it. I think we've come to the end of the time. So again, thank you for a really interesting talk, very exciting technology, and appreciate your time today.
Great, thanks, Chris. Appreciate it.